asthma. As I started out, I mentioned that there were at least two phenotypes of asthma. There's this allergic asthma phenotype, which tends to be steroid responsive. There's a non-allergic asthma phenotype that tends to be steroid unresponsive. A number of years ago, it was thought that mainly Th2 cells, mast cells, eosinophils, and basophils were important in this allergic asthma response, but there are a number of other innate cells that may also be involved in this non-allergic asthma phenotype or pathway that can respond to these non-allergen environmental stimuli. A number of other non-Th2 cells are involved, including innate cells that drive this non-allergic asthma phenotype. Now, importantly, although these pathways can occur independent of each other, it is likely that in a patient with asthma that these pathways intersect. And for example, in a patient who has allergies and asthma, who then develops a viral infection and worsening of an exacerbation of his or her asthma. On the last slide, I'd like to list the people who did all the work. Yajun Chang and Hae-Young Kim are talented postdoctoral fellows in the lab who did all of this influenza work, along with help from other members of the laboratory. Nicole Baumgarth was our important expert in influenza virus who taught us and gave us the influenza virus, taught us how to use this. Elliot Israel and Samsha have been helping us with some human studies with pneumocytes. And Stephanie Shore helped us with the studies with ozone in one of the mouse models of asthma. Andrew McKenzie provided us with expertise in the IL-33 and SD2 pathway, as did Dirk Smith at Andrew McKenzie provided us with the SD2 knockout mice, and Dirk Smith provided us with expertise in the IL-33 SD2 pathway. Thank you very much for your attention. Happy to answer any questions.
Thank you very much. Thank you for a very stimulating presentation, shared very clearly. Can we take questions from the audience here, and then we have perhaps some questions from our video conference site at the Mass General. Questions from the audience?
Thanks for your very nice and interesting talk. I just wonder, you suggest your model is kind of innate lymphoid cell type 2 that is done steroid resistant model, but still just use a lot of that model, and your in vivo model present a lot of neutrophils, not eosinophils. But your data just innate lymphoid cell secret a lot of IL-5 and IL-13. It's kind of not matching, and I just want to more detail about do you see some kind of production of interferon gamma or in that innate cell?
Can you repeat this?
So there are a couple of questions there, but I think one of them was how we can explain the fact that there's no eosinophils when there's still production of IL-5. That's definitely an important question, but I can tell you that we have done studies in the past. Say if you transfer Th2 cells and Th1 cells into a mouse, you can still get airway hyperactivity, but you get no eosinophils. So the migration of eosinophils into the lungs is really inhibited by gamma interferon, and other people have shown this as well. And so I think that's the explanation that we see in this model. But the pneumocytes are definitely producing IL-5 and IL-13, but clearly the IL-13 is required for this airway hyperactivity response. Yeah.
So kind of piggyback on that question. Kind of piggyback on that question. So were you looking at periphery for eosinophils in the model when you were infected with an influenza?
So no, this was in the lungs, in the bowel fluid.
I understand that, but you're saying that interferon gamma in the lungs are preventing the eosinophils from migrating into the lung tissue.
That's correct. That's correct. Preventing them from entering the lungs, so that in the lungs, when Th1 cells are present, as well as Th2 cells are present, there's very few eosinophils in the lungs. In the periphery, it's very difficult to look in the periphery of mice. And so, that's not where we look for eosinophils. There probably are still eosinophils in the periphery, but we don't find them in the lungs.
And I may have missed this. Were there pneumocyte knockout mice that are generated?
So there's no pneumocyte knockout mice. People are working on that now. It's very difficult to find the very specific marker that can be knocked out of these mice, and so that they haven't been generated quite yet. But clearly those would be very useful for a lot of these studies.
Thank you.
Can I just add to the eosinophil bit there? If you take recombinant IL-5 and you put it down the lung of a mouse, you don't necessarily get an eosinophilia. And if you take a recombinant virus such as a vaccinia virus overexpressing IL-5 in a transient manner, you don't necessarily get an eosinophilia. What you need primarily is a second signal from the delivery of an antigen or some other factor in the system to get a pronounced and robust blood eosinophilia and recruitment of eosinophils into tissue.
Thank you very much. I think that helps explain this.
So could you say a little bit about what the sort of innate role of these cells is? Obviously they're bad if you have asthma, at least convince me of that. But what are they good for?
So the question is, what is the evolutionary role for these ILC2 cells, these pneumocytes? Well, the initial studies found these cells in the context of Helminth infection. So they may be important in that role to recognize the presence of worms in the gut and to activate the immune system. David Artis also suggests that in the lungs they produce amphiregulin, which may be important for lung homeostasis, for repair of lung epithelial cells. So that's what has been proposed for these pneumocytes. I think they're like Th2 cells. Why are they there for? And most people believe that they're there for their Helminth infections, and I think that's going to be at least part of the role for these ILC2 cells.
We had promised our colleagues at the Mass General if they had a question they might ask it. You've been very patient. Is there something you want to share, a question that you might ask? Here's a chance. We have you on our monitor screen.
Can you hear us?
Yes, thank you.
This is Johnson Wong. I just wanted to, in terms of your model, the influenza-induced leukocyte activation is not blocked by steroid. How about other models? The leukocytes activated by other triggers, are they still refractory to steroids? And clearly, we do see even in respiratory infection-induced asthma, they still tend to be responsive to steroid. They often need higher dosage, but they're not totally refractory. How you can reconcile with your data?
Right. So those studies that I showed you for the corticosteroids, we were basically using doses that have been shown to be effective for the allergen-induced model. And it's possible that if we use much higher doses, that we could affect some of the pneumocyte function. And so what we believe is that the pneumocytes may not be totally resistant to steroids, but they're relatively resistant. And I think the way you might look at it is really that the Th2 cells and eosinophils are actually very sensitive to corticosteroids, whereas many of these other cell types, and particularly the innate immune system, tends to be relatively resistant to corticosteroids. And that may explain the relative resistance of these innate phenotypes of asthma to corticosteroids. They're not totally resistant, but they tend to be more resistant.
Another question. Pneumocytes seem to play a role in your influenza model, which presumably has a certain amount of lung damage associated with it, as well as the protease allergen model that you mentioned. But they don't seem to have a role in the OVA allergen model. Do you think that pneumocytes could play good, their specificity could be due to lung damage, or is there anything specific about these infection models or certain allergen models?
So I think I heard the question as, since the pneumocytes or ILC2 cells are important in just some models, what is the common feature? I think the most important feature, the most important aspect is what is causing IL-33 to be released or produced. And in the OVA model, I think that the amount of IL-33 is relatively low. And in the influenza model, the macrophages get infected, they produce IL-33. In the protease model, there's damage to the epithelial cells, and that's what releases IL-33 from the epithelium. So I think that I would predict that the phenotypes or pathways that can induce IL-33 production or cause it to be released are going to be the ones where these pneumocytes are going to be important. And in the allergen model, I think there's very little IL-33 released, and that's why the pneumocytes are not required for that pathway.
We're going to take our next question here in the auditorium. Dr. Weiss.
Dale, the two most common viral infections in early childhood asthma are rhinovirus and RSV. And so my question is that what you see here with influenza, would we see the same thing with these other viruses, do you think, or not?
So the question is this pathway also going to be important in rhinovirus or RSV? And those we're trying to study that right now. We don't have the answer to that, but again, the questions will be, is IL-33 released in this situation? And if that's the case, then I think that these pneumocytes, ILC2 cells, will be important, but we don't know the answer quite yet.
The follow-up to that, I think, is that what's characteristic about these viral infections in human asthma is that the attack rate is virtually 100% in all humans. So everybody gets infected with these viruses. Everybody gets infected with the viruses. And what's characteristic is that certain people who go on and develop asthma have a more severe response to the virus. So it's less about the infection per se than it is about the host's ability to handle the infection. How do you think that influences what we're seeing here in the mouse state?
So that's another very important question. Let me just repeat it because you may not have heard all of it. In humans, only some people get asthma with infection. And clearly there's an interaction between genetics and the environment. And so we need to start looking at that in terms of these mouse models to figure out how that may relate to human disease. So it's possible that in humans, if you have lots of pneumocytes present in the lungs to begin with, that then with infection you might trigger these. Or it's possible that there are other factors in specific kinds of humans that allow more IL-33 to be released, things like that. As I mentioned earlier, IL-33 is a major asthma susceptibility gene, as is ST2. I should also point out that the transcription factor that is specific for these pneumocytes, ROR alpha, is also a very important asthma susceptibility gene. So that suggests that there may be explanations of how this is going to be more important in some patients with asthma rather than non-asthmatic individuals.
So doesn't that suggest interrupting IL-33 would be a good way of generating novel therapeutics?
So the question is interrupting IL-33 or possibly ST2 might be a good approach. And there are pharmaceutical companies that have developed anti-IL-33 antibodies and also ST2 antibodies. I think the IL-33 antibody is in clinical trials now. I should point out, though, that IL-33 has many effects. And one report has shown that IL-33 is involved in generating viral-specific CD8 T cells. So one has to be careful by totally blocking IL-33 may cause some problems. And so, but the idea, I think, is correct, as you suggest, is trying to figure out the best way to target these ILC2 cells.
One more question.
I have a very hypothetical question. If you look at the biology of these pneumocytes or the transcriptome of the pneumocytes, do you think they'll point us to any other novel directions that would be important targets for asthma that are not the classical canonical Th2 cytokines?
So the question is, by studying the biology or the genome of these pneumocytes, can we find more targets? And I absolutely think that's correct. And I think that assuming that these pneumocytes are indeed very important and at least some pathways that lead to asthma, targeting them through some very specific means could result in a very useful therapy, particularly for acute asthma, for which we don't really have good therapies outside of corticosteroids, which we use a lot of and may not be as effective as we would like.
Just a follow-up to that question, can you only isolate the pneumocytes from the lung, or are there circulating pneumocytes in the peripheral blood?
So the question is, are pneumocytes also in the circulation? There have been some studies in normal individuals where the number of pneumocytes have been found, but they are very, very low, as opposed to NK cells, the ILC1 cells, which are much higher. And I think this is a reason for why these ILC2 cells haven't been described in the past, because they're very low in the peripheral blood. They are in the lymph nodes of mice that are infected with helminths, and so they are probably some form circulating, but they accumulate in only certain organs.
Is there one more question? Did you want the last question then from MGH? Was there one more question?
Yes.
Here comes.
In chronic illnesses, conditions like nasal polyposis or severe asthma, do you see an expansion of pneumocytes in the blood and or the tissues? And if so, where do they come from?
So the study that looked at nasal polyposis and showed that the pneumocytes are present did not say much about the peripheral blood of these patients. It's possible that in some other instances there might be an overflow of the pneumocytes into the blood, but it just hasn't been studied. It's been looked at, as I mentioned, in normal individuals, and the levels are really quite low. I think all of these questions are very good, and it just shows that there's a lot more studies that need to be done regarding these ILC2 cells, which really were described only 2 years ago and were shown by us only last year to be important, at least in the mouse model of asthma. So many other studies need to be done to confirm this idea, and also to look in people to see how important these cells will be in human asthma and possibly and also in human allergic disease as well.
Terrific. Thank you very much. Thanks so much.